But one of the most important things that you need to remember in terms of research methods with psychology is that psychology is a science. There are methods, or there's a method to the madness, so to speak, when it comes to studying psychology. And psychology starts with a theory. Now, y'all may have heard the phrase, oh, it's just a theory, and that is supposed to imply that that is not an accurate statement, or it's just a theory is the same as an opinion. That is incorrect in science. Okay, in science, theory doesn't equal opinion. Okay, a theory is something that has yet to be disproved. A theory is a coherent explanation or interpretation of facts and observations that have been identified in past studies and has not yet been disproved. So when you hear people say colloquially, oh, that's just a theory, they're thinking hypothesis. A hypothesis is an idea that has yet to be tested. There is something that needs to be discovered or tested, and so you come up with a hypothesis on why you think it is the way that it is. And then what do you do with that hypothesis? You test it. Okay, you perform an experiment. Gravity, for instance, is a theory. Does that mean that when you drop something, it's going to all of a sudden defy the theory of gravity and start floating? No. Okay, so in science, a theory doesn't mean the colloquial misuse of, oh, it's just an opinion or an idea. The theory is evidence-based. It is a coherent explanation of why this works the way that it does. All right, and so this is how you develop a theory. You have to observe what's going on. So I observe that students are happy when they are faced with a smiling teacher, seem to be much more relaxed and much more optimistic, or build an easier rapport if their teacher is smiling and they have that positive environment. So I make a hypothesis. I theorize that friendly teachers make students smile. So for instance, like one aspect of psychology that we're going to talk about is facial expressions and micro expressions, how to tell when someone is genuinely happy or if they're just smiling to fake it. That's called Duchenne smiling, Duchenne, where when it's a genuine smile, the corners of the eyes actually wrinkle or crinkle. You can see like the lines that sometimes people call crow's feet, they become more emphasized. That's when it's more of a genuine smile. So even with wearing masks, you can kind of tell when people are smiling because of how their eyes move. It's not just the mouth that makes that expression for us. So I make a hypothesis, theorize that friendly teachers equals students being happy and them smiling. So how do I put that theory to the test? I have to make a hypothesis. A hypothesis is a specific, testable description of the expected outcome of the study. So you're making a bet, a specific, testable description of the expected outcome of the study. So you really need to emphasize specific, and you need to emphasize testable. Okay, we have to be able to put your hypothesis to a test. We have to make an experiment. So I theorize friendly teachers make students smile. Therefore, I have to make an if-then statement. That is our hypothesis. If a teacher is smiling or friendly or approachable, then students will smile. So a cause and effect relationship in this if-then statement. Once you have your hypothesis, you have to come up with an operational definition for how you're going to measure the variables within this experiment. So your variables are, you have a teacher that's friendly, and then you have the variable of students will smile. So a variable is a thing that can be changed or things that are changed. And you have two types. You have independent variables, the variable that's being manipulated or changed, and then you have the dependent variable, the effect. There is an if, which is the cause, and a then, which is the effect. If is the independent, so if the teacher has a smiling face, then the students will smile. So another theory you could make. I observe that when I drink coffee in the morning, I have more energy. So I theorize that caffeine contributes to my energy. So if I drink coffee, that's my independent variable, that's what I can manipulate, then I will have more energy. That's my dependent variable. That is the effect that I am measuring. And so the methods for researching these hypotheses fall into three different categories. One is descriptive, that describes the event that's taking place. That can include like a case study of an event. Another is correlational, which describes the relationship between variables, and this can be a positive or negative relationship, not in terms of good or bad, but positive as in as one increases, the other increases, or as one decreases, the other decreases, or sometimes as one increases, the other decreases, so it would be the opposite. And that just shows that there's a relationship between those two variables, and can predict, not with 100% certainty, but can predict behavior. But the only one, and you need to write this down and drill it into your head, the only one that actually shows cause and effect, the only one that we can draw causation from, is the experimental method. If you hold an experiment, then you can show causation. But if you show a relationship, like through a survey or just through observation, that's correlational. And it's important to note, causation does not equal correlation. Just because there's a relationship between variables doesn't mean that it shows a cause and effect relationship. There are three different categories of research methods that we use. Today we're going to talk about the experimental method, which is the only one that shows cause and effect. So that is important to note. You do need to write it down and highlight it or put stars by it. The other methods, like correlational, shows a relationship. That means that there could be some sort of relationship between two variables that cause them to increase or decrease, but we can't with 100% certainty determine if they cause each other. We can just see that they are connected in some way. And descriptive research helps with observations that lead to defining those relationships that could exist in order to help predict behavior. So in this lecture, we're going to focus on the experimental method. So an experiment involves manipulating an independent variable in an attempt to determine if a cause and effect relationship exists on the dependent variable. So the independent variable is the thing that you have control over, that you can manipulate, and then you observe the dependent variable to see what changes based on what you did to the independent variable. So, some positives for this method, if you're engaging in research, include that this is the only research method that can identify cause, because in this method you can directly control the variables in order to determine if there is a relationship between them, if it is cause and effect. The negatives, however, is that it's very difficult to replicate real-world conditions in order to show that cause and effect relationship. For instance, if you were trying to show that a certain teaching method leads to increased retention, well, how do you determine who gets that teaching method and who doesn't get that teaching method? Okay, so it's very difficult to account for confounding variables too if there could be other variables that are having an effect on the dependent variable other than the one that you designated as the specific cause. And so it's difficult to try and copy what's happening in the real world and then make it into a clinical experiment where everything goes right. And you also have an issue where some experiments may be unethical. So in terms of if you're trying to show whether radiation leads to a carcinogenic effect, like radiation could lead to cancer, well, it would be very unethical to try and experiment with that on humans, and you're basically trying to give a human cancer in order to prove your hypothesis. So yeah, those experiments would be considered unethical, and thus they would not pass the threshold for being able to put that experiment into play. So what are the parts of an experiment? Well, the hypothesis is your idea of what may be happening. All right, so your hypothesis is what you think is the cause and effect relationship, the if then statement. So if X happens, then Y. Your operational definition, and this is very important, are clear and detailed statements about how your data is measured. And this is important because in every experiment, you need to be able to replicate your results. So you have to give very detailed instructions on how you carried out your experiment so that someone else could do that same experiment exactly the way that you did, and in order to see if they get the same results as you do. Okay, so it needs to be replicable. Okay, so you need to be able to repeat it. So your detailed statements or operational definitions need to define what is the control group, who makes up the experimental group, what are the variables within this experiment, what are the steps in the experiment, and they need to be as clear as possible so that they can be copied and tested over and over again. Then you have your independent variable, which is the thing that you manipulate or change. So if we were using the example of our unethical example of increasing radiation leads to cancer, the independent variable would be the amount of radiation. Okay, the dependent variable is the effect, would be the amount of cancer that did or did not result. You're also, if you're going to have a control group, which means a group that is not changed based on the independent variable, you're going to have to give that control group something in order to make the experiment valid. Okay, so if you have two groups of people and you tell them that you're testing a medicine and they only see group A getting the medicine, then group B is going to think, oh, we're the control group, and that may have an effect on your experiment. So in order to make this a blind design, in order to make sure that the people participating in the study don't inadvertently change the results by knowing whether or not they're in the control group or in the experimental group, whichever group gets the medicine, okay, that's going to be your experimental group, but the control group has to be given a placebo or a fake pill in order to keep the measures as standard as possible. So neither group should know whether they are getting the real medicine that's being tested or if they're getting the placebo that is designating them as a control group. Confounding variables are outside factors that may skew the data. So, for instance, if you're trying to test radiation and whether or not it leads to increased risk of cancer, well, there are other factors that could be leading to someone getting cancer. What if they are predisposed to cancer due to a family history? Or what if they go to the tanning booth a lot, or they go to the beach a lot, and that predisposes them to cancer? Okay, so those confounding variables mess up your ability to determine a cause and effect relationship, because someone could come back and say, well, actually, your experiment didn't show cause and effect between increased radiation leads to an increased risk of cancer, because the people in your experimental group had a family history of cancer on both sides. And so it could be genetic rather than an effect of the radiation that was from their environment. Okay, so you have to determine within your experiment what are some things that could get in the way of proving your hypothesis. A double-blind design is the best design because this is a design in which neither the experimenters, okay, so the people conducting the experiment, nor the participants, meaning the control group and experimental group, know who is in the control group or in the experimental group. Okay, so this is a double-blind design where the people in the experiment don't know if they're getting the real medicine or the placebo, but also the scientists or the researchers who are conducting the experiment don't know who is getting the medicine and who's getting the placebo. And so in order to do that, you'd have to label the medicine with a number or label the participants with a letter so that there is no chance of the people giving the medicine knowing, oh yeah, this is the green bag, that means they're getting the good stuff. And this is the yellow bag, and that means it's placebo. Because you may have an example of hidden bias where The experimenter, because they know, or researcher bias, because they know who is getting the medicine and who isn't, because they know who is in the control group and who is in the experimental group, they may inadvertently and unconsciously send signals that affects the control group and the experimental group. And that's called researcher bias. We're going to talk about that when we get later into the PowerPoint. And so in psychology, this is usually going to be a behavior or a mental process. But it's very, very important that you operationally define your dependent variable. So if you say that the introduction of this pill, let's say that this pill has a lot of vitamin C, that there's a cause and effect relationship between the amount of vitamin C and your intelligence. Okay, so you would say that you are testing the increase or decrease in intelligence as the dependent variable. Well, how do you define intelligence? Well, if you're going to define intelligence, that means that maybe before the experiment begins, you give them an IQ test, which we're going to talk about how that's flawed as well. But you could give them an IQ test before the experiment and then after the experiment, and that could be your measure of intelligence. If you're trying to test kindness, or how would you define kindness, or how would you define a mood change? Okay, so if you're experimenting with, let's say, an SSRI, which is a pill that's typically used to treat depression, how are you going to define depression? If you say in your hypothesis, if a person takes Paxil or Prozac, if a person takes Prozac, then their depression will decrease. Well, how do you measure depression in a person? You would have to come up with an operational definition of what are the symptoms of depression, so that when you're in this experiment, you can notate subject is less depressed or more depressed, or they're showing the opposite of what your operational definition detailed as depression. So it's very important that when you're testing something in an experiment, that you have a definition for it. So with the variables, independent is if in the if-then relationship. It is the manipulated variable, and it is the cause that you're looking to prove in your experiment. Dependent is the then in the if-then statement. It is the affected measured variable. So an example, if you have more books and toys and other stimuli in your environment, will you be smarter? So in 1972, researchers tried to test this. So what is the independent variable and what is the dependent variable? Well, what can be changed here? What can you, as the individual researcher, manipulate? Well, you can manipulate the environment. The books and the toys that are in the environment are your independent variable. So you would create an enriched environment, and then you're looking for the effect that it has. So your dependent variable would be the level of intelligence within those lab rats. All right, so you're going to have to define, how do you measure intelligence in a rat? I mean, you can't give them a multiple choice test or a standardized test and say this is it. And so the way that they defined intelligence in this experiment was, we're going to look at their brain. They found that rats in the enriched environment had heavier and thicker cerebral cortexes, which is your brain, than the rats that were in the impoverished or the environment without books or toys or other stimuli. And so this is how they ethically showed that there's a connection between having books and toys and other stimuli in your environment and the level of intelligence or the level of development of the parts of your brain that are often corresponding with intelligence and critical thinking. Okay, so they couldn't test this on babies because that would be very unethical. How could you give one group of human children more books and toys than the other? That's just rude. And then, in order to test that, you can't just go cutting into babies' heads and looking at their brains. That's also unethical. Okay, so we're going to start talking about ethics in psychology as well in part 3 of our lecture series, which is more self-paced. So clearly the independent variable here is the books and the toys in the environment, and the dependent variable, which they defined operationally by the amount of cerebral cortex left in the rat, that was their dependent variable. So how do you create a perfect experiment? After you define your independent and dependent variable, you need to find participants for your experiment. Okay, now in order to find who is or isn't in your experiment, you need to randomly sample from a representative population. Okay, so for instance, if you're trying to test the relationship between radiation and cancer, you can't make your study entirely out of men because that is not representative of the population at large. 49% of the population is male, yes, but the other 51% is female. Okay, so an experiment that is completely male in terms of who it is using or sampling, that is problematic. So you want to get a random sample, meaning you're not specifically picking out, okay, I want that lady and that guy and that kid or whatever. You need to pick it from a representative population. Okay, so you might say a population from the ages of, let's see, the age of consent for signing on to these things might be 18. So from the ages of 18 to 60, male and female from all different races in the United States. You try to make that your representative population. Once the researcher has their sample, well, that doesn't mean you can test every single person between those ages and then within those standards. You have to pick a smaller amount, a sample from that bigger population. Okay, so once you have that sample that represents the population at large, then you need to randomly assign which people are going to be in your control group that are getting the placebo and which people are getting your experimental independent variable. So you should never self-select the sample or the groups. You should make sure that it is completely random. So put it through a program that gives out numbers or flipping a coin, that sort of random representation. You shouldn't just say, okay, I'm going to put these people over in this group and then these people over in that group, because then you could have even inadvertent researcher bias. So the groups that we were talking about, you have a control group, which does not receive the independent variable, and the experimental group, which does get exposed to the treatment or to the independent variable. And the reason that we have a control group and an experimental group is that we can compare the results of what happens when you do give them the independent variable and then what happens when you don't give them the independent variable. You have to have that comparison in order to show causation. So this control group is the one that's often given a placebo, usually in the form of a pill, so that they don't know that they are the control group, because that could inadvertently lead to skewed results. And there is something called the placebo effect, which basically means if you think it works, then it might. So let's say if you are having a headache and you take ibuprofen, so you know consciously that you are taking medicine, and so that might give you the idea or the predisposed belief that this is going to help you with your headache. So even if, let's say, the ibuprofen is expired or it's not at its most potent level, you might still, regardless, lose your headache because of the expectation that, oh, this ibuprofen is going to help me with this thing. Now that we've talked about what you need to make a perfect experiment, let's look at what you want to avoid in an experiment. You want to avoid confounding variables. These are variables that can distort the results of an experiment. They are hidden or an uncontrolled aspect of the experiment, which is what makes experiments so difficult to pull off in terms of testing every single hypothesis that you could possibly have. One, trying to test every single hypothesis that a person can come up with could potentially be unethical or impossible, but also because there's so many different variables that you can't extract from the situation that it would be futile. So for instance, if you're trying to test intelligence in a group, well, let's say that you have the experimental group in a different room than the control group. Okay, well, if the air temperature is different in one room versus the other, okay, that could have a confounding effect. Y'all may have remembered that earlier, the past couple of years, Mr. Farquhar's room down the hall was bitterly cold. Like, students were bringing blankets and just huddled up. Like, you could have built an igloo in there, is what it felt like. Well, could that have an effect on your test-taking abilities? It very well could. Same thing that if you have the opposite. If you have a sweltering room where it's super hot, that could also have an effect on how you're taking that intelligence test. So if you test the experimental group in one room and the control group in another room, that could mess up your results in that it's not the fact that they received a placebo or that they received the independent variable that led to their intelligence increasing or decreasing as measured by the test. It could be that they had something else distracting them from the test. So, in continuing to create a perfect experiment, you want to set up controls to stop possible bias and those confounding variables. One such control is by creating a double-blind procedure in which neither the research participants nor the staff that are carrying out the procedures know who is receiving the real treatment or who is receiving the placebo. Now, this is done because of the concept of researcher bias. This is where an experimenter's unconscious or unintentional expectation of results may interfere with how they treat the control group or the experimental groups. So while giving the treatment of either a pill, which is a placebo, or a pill that is the actual independent variable, the research staff might inadvertently say like, oh yeah, you're looking better today, or oh man, I know that it sucks that you have to take this pill, but it's important. Well, what if they know that the person they're giving the pill to is getting a placebo? Well, then if the person throws the pill back up and is like, oh, sorry, I couldn't take it, the staff member might say, oh, it doesn't matter. And that would lead the person taking the pill to think, oh, I must be in the control group, because if it mattered, if I were in the experimental group, then they would have just gotten me another pill and made me take it after throwing up this one. So even though that may be unintentional, you still want to try and control those types of biases and try to eliminate them from your experiment. Because often expectations can become reality. It can become self-fulfilling. So, last step for a perfect experiment. You need to check for statistical significance. And now we're going to learn how to do this in our lecture series on statistics. So yes, there is going to be math in this course, but don't worry. If I can do it, you can do it. I'm sure most of you have heard of mean, median, and mode. It's about that level, just percentage-wise. So you want to check for statistical significance, which means that the differences between the groups could not have happened just randomly. So you want statistical significance in order to validate your hypothesis, in order to say, yes, it appears that there's a cause and effect relationship. But even then, even if you get results that appear to prove your hypothesis, you can't stop there. After that first successful experiment, you have to replicate it again with different people to see if you get the same results. And other people are going to replicate the experiment to double check your results. Okay, so it's important that you have those operational definitions so that it can be replicated exactly as it occurred, or as close as exactly as it occurred, but with different people. Some other terms that you need to be familiar with include the term confederate. A confederate is a person posing as a participant, but is actually in on the study. So you may have somebody who is a part of the research staff who goes undercover to be a part of the experimental group or a part of the control group. It depends on the type of study that you're doing. So for instance, when it comes to Milgram's experiments on authority, which y'all should read in the ethics and psychology lecture unit that I provided you, that's self-paced, you would have somebody on the other end of that buzzer who would pretend that they were getting shocked and they were being hurt in order to create the environment that could help test whether or not they were following instructions for the authority. So that person who was a confederate, they were in on the study, weren't actually being shocked in that Milgram experiment, but they were pretending to be so that they could study the reaction of the participants who were pushing the buzzer. All right, so that's an aspect of experimentation known as deception, which can be allowed in an experiment. It is ethical to use deception as long as after the experiment, the participants are debriefed or they're made aware of the deception and they're given more information about the experiment. You also have to have informed consent. So you have to make sure that the person participating in your experiment is not coerced, right? They have to know what the experiment is about as far as you can. Like, you can let deception be in there as a part of that as long as it's explained after the experiment, but they have to know that they will be safe and basically what the experiment will entail. After the experiment, they will have to be debriefed. meaning they're told what you were actually studying if you included deception, and just basically discussing the experiment on a more thorough level. And you also have to maintain confidentiality. Okay, so confidentiality, that the results of this experiment will keep their name and identity private and it won't be used against them in any way. All right, so now what I would like to do is go through some questions, and what I would like for you to do is, as we go through these questions, determine what your answer would be, and I'll give you the correct answer and you can check yourself. Number 1. In an experiment, which group of participants receives the independent variable? If you put answer choice C, you would be correct. The experimental group receives the independent variable. The control group receives the placebo. The population is the population you're trying to test. The sample is the percentage of the broader population that you are testing. Number 2, both the researchers and participants in a memory study are ignorant about which participants have received a potentially memory-enhancing drug and which have received a placebo. What is this type of study called? If you put answer choice B, you would be correct. Double-blind means that neither the researcher nor the participants know who is in the control and who is in the experimental group. Number 3, a researcher looking to determine a cause and effect relationship between two variables should choose which research method? If you put answer choice D, you would be correct. Experimental is the only one that shows cause and effect. Correlational shows a relationship but does not show cause and effect. And naturalistic observation and case studies are a part of descriptive methods. Number 4, the variable manipulated in an experiment is known as the what? If you put A, independent variable, you would be correct. The independent variable is what you, as the researcher, can manipulate. The dependent variable is the thing that you're trying to measure that changes as a result of the introduction of the independent variable. There's no such thing as a control variable, and an experimental variable is simply either independent or dependent or confounding.